everyone, I am Anshit. I have studied in Cambridge High School. I have secured 93% in SSLC examination. My aim is to become an engineer. I have chosen Cambridge PU College of Science either because the lecturers are very well experienced. The atmosphere is very well and very organized. I request you to visit Cambridge PU College of Science. Thank you.
Myself, Vijayalakshmi. I am from Cambridge Primary and High School, Bidar. My 10th percentage is 97.60. I took my admission in Cambridge PU College of Science. Here I like the learning atmosphere, high teaching faculty. I love the teacher's explanation.
Hello everyone, myself Divya Jyoti from Maulana Azad Model School, Ratkalpura. I have secured 80.64 percentage in my 10th standard. So I have took admission in Cambridge group of institutions.
Myself Ananya. I am from Sri Vidyaranya High School. My 10th percentage is 88 percentage. This campus was very good. Lecturer's explanation is very nice. So I took admission in Cambridge PU College of Science. I want to become a MBBS doctor.
Let's look at the simulation of gene expression in a body. As you know that for every character, there is a gene within the body in the DNA. So like for example, if I say like eye color or hair texture, whether it will be curly or whether it will be smooth, or the skin color, for every character, somatic character in the body, there is a one gene in our DNA. So let us see how it works based on with the help of the simulation here. So you can see for gene expression, we need DNA. As you can see, the DNA is double stranded here, and then there is a regulatory protein. I mean, like for this area which it is showing, it is transcribed region. For gene expression to occur, there are actually 3 steps. That is, first it has to be replicated, then it has to be transcribed, and then it has to be translated. So you can see this is a double stranded DNA here, and I am just trying to show you with the help of simulation that how the gene expression occurs. So taking just like you consider this one as a first gene here, and you know for a gene expression, transcription and translation both are needed. So what is transcription? It is the formation of RNA from DNA. And what is translation? That is the formation of protein from the RNA. So let us see how it works here. Now you can see already the DNA is here, and this is the region where it has to be transcribed. Now considering it as the first gene, let us see how it works. So for effective gene regulation, there are some regulatory proteins also. Like for example, this regulatory protein which is there in the regulatory region, which is needed for effective gene expression, and definitely for transcription to occur, we need a RNA polymerase. Now you see how it is happening. It is moving through the DNA, and here you can see the RNA has been formed, and now here you can see now you see how here the mRNA is forming the RNA. It is being formed here, and now you know, like for translation we need what? We need the ribosomes, the smaller subunit and the larger subunit. Now you see how the protein has formed. So you can see this protein has formed here, and since the gene is same, so same protein is forming here. But if we go for the another gene, now you see the same process how it is continuing. Again, if we take this regulatory gene here, and this is the RNA polymerase, and we need some positive transcription factors here. Now you see this mRNA polymerase, if it works on the DNA, see how it is moving and how the RNA is being transcribed here. As it moves here, see this is the RNA. Now for protein synthesis, as I said, it needs a ribosome. It needs an association with the ribosome, and see the another protein has formed here. See, this is the protein which is being formed here because I have taken the next gene. So like that, many genes, they continuously, it will be going in a commaless fashion, and most of the genes, all the genes are transcribed till we get the termination factor. And for, you know, like translation, there are genetic codes are there. Unless they encounter the stop codons, they'll keep continuing the translation process. So similarly, you can go for the next one. Now you can see there are some negative transcription factor. Like since regulatory means what? Either it may be on the positive side or in the negative side. Let us see if it is a negative transcription factor, then what may happen here. Now this has come. Now see, the transcription is not going on here. Why it's not? Because the regulatory, in the regulatory region, the negative transcription factor has come. So it is not allowing the gene expression. So by this video, we can see how the gene expressions takes place by the replication, transcription and translation. Now, so like this, based on what kind of gene is here, we can, it is decided in the cell that whether what kind of proteins will be formed. If it was a gene 1, one kind of protein will be formed. If it is the next gene, the another kind of protein is formed, and the another for the next gene, like I have shown just now. So like that, for different genes, different kinds of proteins are formed, and based on these proteins, the body characters are expressed. Like, thank you so much.
Okay, let us start this simulation as based on Bohr's atomic model using the Planck's quantum theory. As this is have a different cell as structure is a modification. As here center have a nucleus and revolving electrons are revolving around the nucleus. Suppose when we falling of hydrogen light on this particular structure here, the electrons it will moving around the nucleus in a definite path that we called as cell or orbit, having a different cell as this one is called as K cell, this is L cell, this is M cell, this is N cell, this is P cell. A various cell it will represented as 1, 2, 3, 4, 5 respectively. Electrons are revolving in its own path that we call as orbit or cell. Whenever electrons are moving around the nucleus, it does not lose or gain the energy. It means energy remains constant. As another thing, whenever those cells are nearer, as closer to the nucleus, they are having the more energy, and because of lot of attractions is occurs in between the nucleus and electrons. Then another thing is those cells are away from the nucleus, far distance, so they are having the less energy because of less attraction in between the center of nucleus and outermost cell. As come to the third point, as here whenever the electrons are jumps from outer cell to the inner cell, so then the emission of radiation is occurs that we called as E1 is inner cell, E2 is outer cell. From outer cell to inner cell, that is E2 minus E1, it shows h nu, nothing but photons. And another thing exactly vice versa, whenever the electrons are jumps from inner cell to outer cell, that we called as absorption of photons, then that means E1 minus E2 is equal to h nu. H nu is nothing but photons, h means Planck's constant and nu is frequency of radiations. See here, whatever the wavelength it required, whenever white light is falling on this structure of atom, so here this region is nothing but UV region, and its wavelength is starts from 380 nanometer to 780 nanometer. On this region, the different color is formation takes place, as this is the red colored as light is falling on this particular structure. So then it will shows the different region. See here, have different transition based on using the different wavelength that is measured in nanometers, and their transition have a different range, as from first cell to 6 cell, from first to 5 it requires 95 nanometers, as like different cell transition, it requires the different wavelength. Simple conclusion, whenever increasing the wavelength, so then here the cell number is also away from the nucleus. It means 2 to 4, 486 nanometer, again away from 3 to 4, again above the 1000 nanometer. As here, this is the electron energy level. As energy level, this is the ground level. Electrons are jumps from ground level to the higher level. That shows n1 is equal, n is equal to 1, 2, 3, 4, 5, 6. These are indicates as the first cell it is Lyman series, Balmer series, Pachand series, bracket series and Fund series. Total have a 5 series, 5 lines it will shows. By using the hydrogen atom, it occurs in a different lines. It will occurs. This is the Bohr's model of an atom. Finally, it is observed by using the spectrometer, and that clearly we observe how much of lines it will occurs from the hydrogen atom. Thank you.
Okay, students, let us have a look at the projectile motion, the simulation of projectile motion. यहाँ पे देखिए, ये हम नॉर्मल प्रोजेक्टाइल फायर करते हैं, ये है एंगल ऑफ प्रोजेक्शन. यहाँ पे देखो, ये है एंगल ऑफ प्रोजेक्शन, एंगल ऑफ प्रोजेक्शन. जब कभी कोई प्रोजेक्टाइल मूव करता है तो इसका जो एक्सेलरेशन है वो बस एक ही डायरेक्शन में होता. ये देखो एक्सेलरेशन, ये वाला नीचे है. एट एनी पॉइंट अगर मैं एक्सेलरेशन देखूं तो इसका एक्सेलरेशन ऑलवेज डाउनवर्ड्स, एक ही एक्सेलरेशन है इसमें, एक्सेलरेशन इन वाई डायरेक्शन ओनली. ठीक है, इसके अंदर ए में एक्स डायरेक्शन में कोई एक्सेलरेशन नहीं है, नो एक्सेलरेशन इन एक्स डायरेक्शन. नो एक्सेलरेशन इन एक्स डायरेक्शन. और वाई डायरेक्शन में एक एक्सेलरेशन है जो कि नीचे की तरफ है, जो माइनस जी होता है. ठीक, और ये जो मैक्सिमम, इसका मैक्सिमम हाइट है एच मैक्स, इसको बोलेंगे एच मैक्स, मैक्सिमम हाइट रीच किसी भी प्रोजेक्टाइल का. फिर ये जो डिस्टेंस है ना यहां से यहां तक जहां पे प्रोजेक्टाइल जाके गिरा, इसको बोलेंगे हम रेंज. रेंज मैक्सिमम कब होता है? मैक्सिमम रेंज आर मैक्स, मैक्सिमम रेंज होता है एट प्रोजेक्शन थीटा इज इक्वल्स टू 45 डिग्रीज पे. यहाँ पे हम देखेंगे, ये अभी मैं 50 डिग्रीज पे प्रोजेक्ट कर रहा हूँ, फिर 45 पे भी प्रोजेक्ट करके देखेंगे और देखेंगे कि मैक्सिमम रेंज कब होता है. यहाँ पे मैं इसको 50 पे प्रोजेक्ट कर रहा हूँ, तो 50 पे प्रोजेक्ट करे तो ये कहाँ पे जा रहा प्रोजेक्शन, ये यहाँ पे नीचे की तरफ जा रहा है, ये डिस्टेंस पे अभी ये है. और मैं इसको अगर एंगल थोड़ा सा कम कर दूं 45 डिग्रीज पे, तो ये जो है ना प्रोजेक्शन ज्यादा दूर जाएगा, इससे थोड़ा सा ज्यादा दूर. और मैं एंगल और कम कर दूं इसका 35 डिग्रीज, तो ये और कम डिस्टेंस पे जाएगा. और अगर मैं इसको एंगल इंक्रीज कर दूं 65, तब भी इसका रेंज कम हो जाएगा. तो रेंज मैक्सिमम कब होता है? एंगल 45 डिग्रीज पे. नेक्स्ट देखेंगे हम वेलोसिटी वेक्टर्स, यानी कि वेलोसिटी किस डायरेक्शन में होता है और किस किस कौन से वेलोसिटी वेक्टर्स हैं. अब जैसे कि कोई एक अगर प्रोजेक्टाइल जा रहा है तो उस प्रोजेक्टाइल का वेलोसिटी दोनों डायरेक्शन में होता है. यहाँ पे इस डायरेक्शन में भी इसकी वेलोसिटी होती है, आगे डायरेक्शन में भी, और ऊपर डायरेक्शन में भी इसकी वेलोसिटी होती है. इस पॉइंट पे सिर्फ एक ही वेलोसिटी होती है, आगे वाली, जो कि एक्स वाली वेलोसिटी, एक्स वेलोसिटी रहती है. और जब यहाँ पे होता है तो इसकी वेलोसिटी एक नीचे और एक आगे वाली. तो आगे वाली जो वेलोसिटी है वो हमेशा सेम होती है. ठीक, अब आगे वाली वेलोसिटी को यहाँ से अगर कोई एक ऑब्जेक्ट में प्रोजेक्ट करा इस डायरेक्शन में, और इसके दो वेलोसिटीज हो गए, ये वाली वेलोसिटी हो गई वी वाई और ये वाली वेलोसिटी हो गई वी एक्स. तो अगर ये इनिशियल वेलोसिटी से हम प्रोजेक्ट करें u, तो इसकी x वाली वेलोसिटी हो जाती u cos थीटा और ये y वाली वेलोसिटी हो जाती u sin थीटा. अगर थीटा यहाँ से दिया हुआ है, तो एक एग्जाम में ऐसा भी आया था, थीटा इधर से दे दिया था, तो उस केस में होता y वाली वेलोसिटी इसकी u cos थीटा होती और इसकी x वाली वेलोसिटी u sin थीटा होती. ठीक, हम देखेंगे इसके वेलोसिटी वेक्टर के कंपोनेंट्स और कहाँ पे वो वेलोसिटी कम होती है या ज्यादा होती है. देखेंगे हम इसकी, तो देखेंगे ये पूरा हम इसको रीसेट कर देंगे. वेलोसिटी वेक्टर्स देखेंगे, ये वाला वेलोसिटी वेक्टर है, एंगल ऑफ प्रोजेक्शन अप्रोक्सीमेटली 50, ये स्पीड थोड़ा सा इंक्रीज कर लेंगे 21. तो देखो इसको केयरफुली देखेंगे हम. यहाँ पे ये पॉइंट पे सिर्फ आगे वाली वेलोसिटी है इसकी, और ये पॉइंट पे ये इसके डायरेक्शन है. अगर इसको कंपोनेंट्स में हम सेपरेट करेंगे तो ये इसके कंपोनेंट्स अगर सेपरेट करेंगे तो ये वाले और ये वाले दोनों कंपोनेंट्स हमको दिखेंगे। ठीक है, इसके अगर मैं एयर रेजिस्टेंस इसको कैंसिल कर दूं, एयर रेजिस्टेंस तो थोड़ा सा आगे जाएगा ये, और अगर इसके मैं कंपोनेंट्स देखूं तो ये वाले कंपोनेंट्स आपको नजर आएंगे। ये x का कंपोनेंट हो गया और ये y का कंपोनेंट हो गया। इसको हम स्टार्टिंग से हम ये करेंगे, प्रोजेक्ट करेंगे। ये देखो x का, ये एयर रेजिस्टेंस नहीं है तो बॉल थोड़ा सा आगे चला गया, और इनिशियल वेलोसिटी मैं थोड़ा सा कम कर दूं तो ये विदाउट एयर रेजिस्टेंस है। ये कंपोनेंट्स देखिए दोनों पॉइंट पे, यहाँ पे इस पॉइंट पे इसकी आगे भी वेलोसिटी है, ऊपर भी वेलोसिटी है, और ये जब मैक्सिमम यहाँ पे पहुंच रहा है, इसमें जो नीचे वाली वेलोसिटी है वो जीरो हो रही है, सिर्फ आगे वाली वेलोसिटी रह रही है। जैसे ही वो नीचे आ रहा है, अब दोनों डायरेक्शन की इसकी वेलोसिटी है। तो ये वेलोसिटी वेक्टर्स है, और एक बार केयरफुली हम देख लेंगे वेलोसिटी वेक्टर्स। यहाँ पे देखो वेलोसिटी वेक्टर, यहाँ पे ये हमको केयरफुली देखना है। वाई वाला यहाँ से जब वाई जा रहा है तो वाई वाली जो है ना ऊपर रह रहा है, फिर जब यहाँ से यहाँ जा रहा है तो वाई वाली जो वेलोसिटी है वो नीचे के डायरेक्शन में। उसी तरीके से अगर हम एक्सेलरेशन वेक्टर्स देख लेंगे, इसका एक्सेलरेशन वेक्टर्स और उसके कंपोनेंट्स, तो ये देखेंगे एक्सेलरेशन वेक्टर्स। एक्सेलरेशन एक ही डायरेक्शन में है, इसके एक्सेलरेशन सिर्फ नीचे के ही डायरेक्शन में ही है। ठीक है, ओके स्टूडेंट्स।
So generally plants are autotrophs. They can prepare food material themselves. So in plants they contain leaves. The leaves are green color because they contain chlorophyll pigment. So when you see the plant cell structure, in this plant cell structure, the outer layer of the plant body is called cell wall. The outer layer of the plant body is called cell wall. Cell wall, it gives definite shape of the plant body. It is a rigid structure. It is rigid structure. So the cell wall is made up of cellulose, and inside the cell wall they contain cell membrane. Inside the cell wall contain cell membrane. This cell membrane made up of phospholipids. Phospholipids. It is a bilayer. It is a bilayer. Next, when you see inside the plant cell, the fluid part inside the plant cell is called cytoplasm. Cytoplasm. Next, inside the cytoplasm, we can see different types of cell organelles are present. So in these cell organelles, main nucleus, endoplasmiculum, Golgi complex, mitochondria, chloroplast. All cell organelles are present inside the plant cell. So in this plant cell, the important cell organelle is called nucleus. It is called nucleus. So this nucleus, in 1831, the nucleus was discovered by the famous scientist Robert Brown. So when you see the nucleus contains different parts like nuclear membrane, nucleolus, nucleoplasm, and chromatin materials. So when see inside the nucleus, there is a small thread-like structures. Those thread-like structures called chromatin materials. During cell division, all these chromatin material undergoes condense to form chromosomes. And after that, when you see in the plant cell, we can see a very large part. This part is called vacuole. So in this vacuole, it is surrounded by layer. The vacuole card is surrounded by layer. This layer is called tonoplast. The vacuole surrounded by layer is called tonoplast. In this tonoplast, they contain fluid. The fluid is called cell sap. And after that, in the plant cell, we can see the green color cell organelles. The green color cell organelle is called chloroplast. So this chloroplast main function to prepare the food material with help of photosynthesis. And in the plant cell, you can see there is another cell organelle. This cell organelle is called mitochondria. So the mitochondria, it is also called powerhouse of the cell. So the mitochondria helps to gives energy. That energy gives in the form of ATP. So that's why mitochondria is called powerhouse of the cell. Next, in this, the nucleus is surrounded by specialized structure that is called endoplasmic reticulum. Endoplasmic reticulum. So this endoplasmic reticulum, it is commonly called cytoskeleton of the cell. The endoplasmic reticulum, it can be divided into mainly two types, RER and SCR. RER and SCR. RER means rough endoplasmic reticulum and SCR means smooth endoplasmic reticulum. So the ER again divided into two types. One, RER, SCR. RER means rough endoplasmic reticulum, SCR means smooth endoplasmic reticulum. So the main function of RER. So when you see the in slide, the RER attached to a small structure. The blue color structures is called ribosomes. Those are called ribosomes. Ribosomes. So the main function of ribosomes are involved in protein synthesis. So the RER, it contains ribosomes. That's why the RER main function protein synthesis. And the SCR in that region. So these are called SCR. In the SCR, ribosomes are cannot attach. So the main function of SCR, lipid synthesis. So different cell organelles are present inside the plant cell. Next,